Can you guys confirm if I'm audible to you and if the screen is visible to you? Am I audible?
Audible, ma'am, but screen maybe my tower is slow. It's not visible.
Okay, right. Let me then reshare. I hope screen is now visible to both of you.
Yes, ma'am.
Yes? All right. Okay. So, if any one of you is joining this today's session for the first time, let me give a bit introduction about myself. I am Mauchandra Paul. I am a PhD scholar at IIT Kanpur in the Department of Chemical Engineering. And in my thesis, I have been working in the field of tissue engineering, where the use of biomaterials uh, is prominent for making various scaffolds onto which cells are grown and artificial tissues are made. So if you want to know more about me and what type of research I do, so you guys can check out my website. And if you have any technical queries on this course, on any other topic of this course, then you can definitely ask me directly during the session that we are having, or you can post it in the course discussion forum. There also, I see the queries and try to answer it to as much accuracy I can. So we are like meeting. So this is the fifth week. So we have been meeting like this for the past 4 weeks, every Saturday from 4 p.m. to 6 p.m. The link is with you, right? The same link is used every week. And the recording of the lectures that we have every Saturday is uploaded in my YouTube channel. You guys can go and watch it afterwards, or those who have missed can also access it. The notes that I use for these lectures are also available, and it is there in the NPTEL login page, right? And I also note the attendance of who all are present, and that's it. And the link that you have used to join this meeting and the interface that is present in front of you, it doesn't have a chat option. So if you want to ask any queries, you can either raise your hand, all right, so that I can call upon you and then you can unmute yourself, or you can directly unmute yourself. There is no judgment here. If you have query at any point of time, you just simply unmute yourself and directly ask me. I'll able to help you as much as I can. Okay. So let's begin today's class with this particular question. So this question is based on the biocompatibility concept. So the earlier weeks of this course, you guys were introduced with the term biocompatibility. But again, this is a very important topic. I want to again slightly revise on what do you mean by biocompatibility. So those of you have understood, all right, can try to answer it. Okay, find the answer with me. If you have not understood the concept of biocompatibility till now, then this question might help you in that, in gaining a better understanding. So in this question, what you have to do, there are five parts basically, and you have to tell which of these parts, or in which of these options, the biomaterial being used is biocompatible. Okay, so let's look at the first part. A woman with breast implant. Okay, I'm reading this part. A woman with breast implant experiences localized swellings in her breast after 1 year of surgery. Now you have to tell me whether, so here the biomaterial that is being used is what? Anyone? You can simply unmute. It's totally fine. So in part A, so this first part, what is the biomaterial that is being used? It's the breast implant, right? The silicone breast implants that become important for those women who undergoes surgeries for breast cancer and all. So this breast implant, because of this breast implant, all right, this woman is experiencing some swellings like edema kind of soft tissue swellings. So now in this case, would you say this breast implant is biocompatible or not? If biocompatible, then we have to tick. If it is not, then we'll move to the next part. Is the breast implant here biocompatible? Can you call this breast implant biocompatible?
No, ma'am.
Yes, you're correct. This is an, so breast implants are supposed to last for 10 years, 20 years, all right? So they are kind of a permanent kind of biomaterial. So if within 1 year of surgery, it because of say any reason, it is it is causing discomfort to the patient, then it is not a biocompatible material. Next part we'll see. So this one. There's a bedridden patient with urinary catheter. Okay, and he is passing clean and clear urine for 2 weeks. So in this case, what is the biomaterial being used on the patient? You guys can try. It's totally fine if you're wrong. Is urinary
Catheter.
catheter? Yes, yes, you're correct. It is urinary catheter. So this urinary catheter, doctors have to insert in those patients who undergoes such kind of surgery that makes them bedridden. Means they cannot go to washroom on their own, and to like filter out their blood, that is to pass urine, they use catheters, and these catheters, through this catheter they urinate on a bedpan or something like that. Okay, so and these are very temporary kind of thing. So when once the patient the surgical area is healed and he is able to walk again, then the catheter is no longer needed. So a 2 weeks time is a very short time to be used. And since urinary catheter are direct in contact with urine that contains a large amount of bacteria and all, so the biomaterial is such that it is resistant to biofilm formation, then it will not cause any infection to the urinary bladder or surrounding area, all right, of the patient. So because of which the urine, like the color of the urine, okay, the turbidity of the urine can get affected. So now in this case, the patient is passing clean and clear urine, means it's transparent and there is no suspension in it. So now in this case, would you say the urinary catheter is biocompatible?
Yes, ma'am.
Yes. So this is biocompatible, and part A, it was not biocompatible. So there was, right. Okay. So this is biocompatible. Now in part C, there's a patient with orthopedic rod inserted in his tibia bone and can do physical activities for several years. Now, without much explanation, I think you guys have understood what I'm trying to say here, right? So, based on your understanding, tell me if the orthopedic rod is biocompatible or not. Read it again yourself and answer. Is it biocompatible? Yes or no? Those who have joined today, would you like to answer? Santosh has been coming for past 4 weeks, so I know that he can. But what about others?
Biocompatible.
Yes, you're correct. Now, in the next question, there's a patient with history of arteriosclerosis. So, what is arteriosclerosis? It is a disease where the veins and arteries, the endothelial lining of veins and arteries, there cholesterol deposition takes place. So, the LDL, low density lipopolysaccharides. Okay, so the cholesterol, that is the cholesterol. There are two types of cholesterols, LDL and heavy, VDL and HVDL. Okay, so for our body, LDL is the most damaging type of cholesterol. If that level is high, then the condition of atherosclerosis increases, because this particular type of cholesterol, it can go and accumulate inside the walls of your veins and arteries and clog it, and basically give resistance to proper flow of blood. So this patient is that kind of disease, like he had that particular disease. Now, so the treatment of atherosclerosis, what people do, they try to remove that cholesterol, right, from the veins and arteries. So for that, they use stents. So stents, vascular stents, are inserted through endoscopic surgery, inserted at the site of clogged artery, and there it, using a balloon, it is inflated. So due to inflation action of the vascular stent, the diameter of the blood vessel, it increases, okay, and it restores the normal flow of blood. Now, if the stent can be supplemented with drugs, those drugs can directly have action on breaking down this cholesterol plaque, you can say plaque, and remove it. Other than that, patient is also given many kinds of medications. Those again do the same action. They remove that plaque from the site of cholesterol deposition. Now this person, he has had atherosclerosis, and because of that, he had a vascular stent inserted in one of his arteries. Now suddenly he experiences a heart attack. Now in this case, this vascular stent, which is the biomaterial here, would you say it is a biocompatible one or not? Now, can you understand why it must have caused heart attack? If then you can definitely answer. Think over it. So the vascular stent that is inserted to make the diameter of the artery larger in this patient with atherosclerosis, is it a biocompatible material? N- No, right? It is not biocompatible. Why I'm saying no? Because this vascular stent, so the reason of heart attack is because the blockage of artery due to deposition of cholesterol plaque. So this vascular stent, the purpose of that stent was to uh, make sure that the plaque deposition doesn't happen. It should ensure smooth, continuous flow of blood. Now it has failed in doing it. Okay, and on the top of the stent, new plaque got deposited, new cholesterol molecules it got deposited, because of which again the artery blocked, and that gave rise to interrupted blood flow and then heart attack. Am I correct? Have you understood why this is not a biocompatible material in this case? Okay. Now, the last part here, the patient is patient, he is having breast cancer, and he or she is treated with the drug-loaded polymer nanocapsule formulation. Now, this formulation this nanocapsules are basically drug delivery systems, and they are made up of a polymer. Drug in this case is not the biocompatible drug, is loaded inside the polymer capsule. Now this polymer capsule is our biomaterial. Okay, this polymer capsule is our biomaterial. Now why they do this kind of that, why they make this kind of drug formulation? To deliver the drug to the correct region in the body, targeted region in our body, and so that maximum benefit can be extracted from that particular drug. So this patient, that's why is treated with this kind of a drug-loaded polymer nanocapsule formulation. Now this patient develops a liver disease. Okay. Now in this case, tell me, is this polymer nanocapsule? Here is our biomaterial. Is it a biocompatible one? Think in this way, has this been successful in achieving its um, it was intended to treat the breast cancer, so has it been successful in doing it? If yes, no, then you will have your answer.
The the biomaterial had done this work right now, ma'am.
So what is your answer? What are you saying? Is it biocompatible or not?
Um, Confused, ma'am.
Okay.
Had this
Biocompatible.
one.
Okay, so the correct answer here is it is not. Why I'm saying this? Uh, this type of nano formulation or this type of drug formulations are made for targeted drug delivery. So ideal cases, it should target the drug only, it should target or should make the drug available at the targeted site at the highest concentration. Okay, so don't think about anything else. Like maybe the person had a liver disease history, or the person may be taking some other medication. Don't of scenarios. Just think the impact of this particular biomaterial in this case on the application, or has it been able to successfully achieve that application or not. That will only give you the answer. Don't think about anything else. So usually what happens when people use this kind of polymer nanocapsules, they make it using some biodegradable polymer, so that they don't so that it can easily pass through uh, the metabolism in liver. So most of the drugs or anything that you eat goes to liver first, and there it is metabolized, and then it is, if it is good, then it will be absorbed back into the blood. But if it's a waste thing, then it will go back and it can either pass through your urine or it can pass through your feces. Now in this case, the liver, assume the liver was completely normal. Okay, so the breast cancer and liver disease, they are not related at all. Now, whatever drug or whatever polymer nanocapsule, it may have happened that it didn't, the drug, it didn't completely reached the breast cancer, or the breast where the cancer is. It didn't reach there at the highest percentage. Some part of it retained in the liver, and maybe the liver was susceptible to that drug. Okay, and because of which there is some malfunctioning in the liver, like function, overall function, and that is why a disease was developed. Now if you think in this manner, so we are focusing on the impact of the biomaterial on the application. Okay, and has it been able to successfully do its application? Now if you read the statement again, you will realize no, rather than solving one problem, it created another problem. That is why this particular biomaterial is not biocompatible. Got the logic? Okay. So I think uh, if you have any confusion in understanding what is biocompatibility, this question must have helped you. But since biocompatibility itself is a very big definition, people have their own meaning of biocompatibility. But if you go through Professor Doble's lecture, there he has mentioned very nicely some bullet points that helps you understand what is biocompatibility. So you can refer those for now. But when you move forward in your research, if anyone wants to do research in this field, then your definitions might change. But the basic thing is the quality of the patient's life should not be compromised due to your biomaterial. It should do the function for which it was designed. Okay, it should not cause unnecessarily uh, problems for the patient, right? So and it should like stay, it should do consistently its function for the period it was intended to. So these are some simple definitions, simple ideas behind the meaning of biocompatibility. Okay. Now, I'll briefly discuss how does our body, that is we are the host, how does our body responds when a biomaterial is introduced inside our body. Okay, I'll briefly discuss that part. So, whenever a surgeon is trying to insert a biomaterial inside your body, they usually do it through surgery. Okay, so I'm talking about those biomaterials which will be inserted inside our body, not those like wound healing patches or uh, inserted like through like feeding tubes and all. So those do not cause injury. Those are meant to not cause injury. I'm talking about those biomaterials which are inserted inside our body. So for doing that, the surgeon has to create a wound and will create an injury, and because of which your biomaterial will come in contact with body's blood, and there will be blood material interaction. So blood contains all sorts of immune cells, right? The neutrophils, eosinophils, then your monocytes, macrophages. So they don't like anything foreign, right? Biomaterial is foreign to our body, although we introduce it to help our body to heal, but it doesn't like it at all. It considers everything foreign except his own cells. That is the normal response. So they will now try to prepare itself. These cells, these immune cells will try to prepare itself to fight this biomaterial. They will like to try to degrade it. They will try to remove it from the body. That is their role. So when the blood, and in the blood, the immune cells come in contact with the biomaterial, then the real game begins. Okay. First, there will be a matrix formation, provisional matrix formation. Here, what happens? Here, your due to injury, your blood will be exposed to air and all, so it will coagulate easily, and it will with it it will trap all the platelets. Okay, these platelets will become activated. They will start secreting factors to start the coagulating factors. It will again, it is like a cycle. Coagulation should happen, otherwise the blood will continuously flow out. Then comes the complement system activation. So there are some proteins called opsonins. So they goes and binds with the biomaterial, and they are get recognized by the immune cells, like the monocytes, neutrophils. So the interaction of these cell types, and they subsequently they release some proteins as well, and because of which they make a matrix surrounding the material. This matrix also helps in migration of cells, right? It provides a substrate. So then starts the acute inflammation part. So again, it is like the involvement of, so up till provisional matrix formation, the immune cells, They were just arriving at the site of biomaterial. They didn't do anything yet. Now their job begins. Okay. So first, what will do? They will secrete some cytokines to recruit more and more immune cells, and they will call upon the monocytes, the macrophages. So the role of macrophages, what they do, they try to endocytose any foreign material, okay, and they degrade it and like flush it out. Okay, our lymphatic system is there where the waste and all gets removed from the body. So the macrophages here are not very active, okay, but similar types of cells, these are called phagocytic cells, the neutrophils, they also try to phagocytose. So in the acute inflammation, the role of phagocytic cells like the neutrophils, and then subsequently the monocytes, and then later on in the chronic inflammation stage, the macrophages. So these all cells, they are phagocytic in nature, and they recruit more and more cells of their own population and try to phagocytose the biomaterial. Okay, now if the biomaterial is small enough, like a nanoparticle, it will get phagocytosed easily. But if it's a very big biomaterial, like your silicon breast implant, or your orthopedic rod or screw, then it is not possible for these cells to do it. So the response continues. The response continues up till a chronic inflammation. So chronic inflammation lasts for months to years. Okay, and if it is longer than that, then sometimes it is maybe due to some infection, some bacterial infection, and it is very very easy for the bacteria to grow on the surface of the biomaterial, because what happens, our body becomes so focused on eliminating this particular biomaterial that uh, it doesn't give enough attention to the bacterial cells which are also there. They sometimes remains hidden behind the biomaterial. Okay, so that is what happens. And over the period of time, these bacterial cells proliferate themselves and create a biofilm over the biomaterial, and then it worsens. It doesn't help, rather it worsens the inflammation process. Okay, the site doesn't heal. Now, suppose after chronic inflammation, like the healing, the surface of the wound has healed up till now, and like your biomaterial, which is still under the influence of the immune cells, it is still going on. But some phase of healing starts to appear when the granulation tissue forms. So what is granulation tissue? So if any one of you have ever observed a small wound onto your skin, and by curiosity have removed that black plaque, so that was your blood clot that was formed initially when the injury happened. So when you remove it, you might see some pinkish layer. It is membranous and very pinkish. So this is the granulation tissue. So granulation tissue is formed due to proliferation of the endothelial cells. So due to damage, due to injury, your blood vessels and all, they are got broken, right? So new blood vessels starts forming at the site of the injury. Then if the surgeon has implanted the biomaterial in skin, then the native skin cells will start to proliferate and cover up the area. So when this thing happens onto like in the presence of biomaterial, when these things happen in the presence of biomaterial, then a granulation tissue forms. But remember, the immune cells are still fighting. Okay, it is still fighting to remove the biomaterial from the body because it is foreign. Okay, so after, or you can say not after, simultaneously, what the immune cells are trying to do, they are trying to increase their strength. How they do that? Many of the macrophages, they fuse with themselves and create something called foreign body giant cells. So these giant cells are multinuclear, as you can guess, and they are much larger than your typical cell size. So they try to engulf, they try to phagocytose the biomaterial. And this response can now continue for years. Okay. So now body thinks like बहुत हो गया, like it is, we are not able to defeat this particular foreign material. So what to do? Uh, and during this immune response, whatever chemicals, what are cytokines and all that are released, they are actually toxic. Okay, that can damage your healthy tissue. So then the body tries to cover it. That phase is called fibrosis, where the fibroblast kind of cells, which are known to synthesize a large amount of collagen, elastin, etc. So the amount of collagen increases, and they create a network, a shield, okay, that isolates this biomaterial with the foreign body reaction from the rest of the body. So it is like the body is still fighting, but the rest of the body can function normally. Okay, so sometimes what can happen, the biomaterial, if suppose if it is orthopedic rod, then fibrosis is fine because its function is just to give strength, right, to the person so that he can walk. But if it is a biomaterial like pancreatic cells, which are encapsulated within some porous scaffold, and now if fibrosis happen on top of that, so then the release of insulin cannot happen. So when fibrosis then the fibrosis due to fibrosis, the function of the biomaterial got completely eliminated. It is no longer useful to the body, right? So the fibrosis is very, very bad, and it can again compromise the biocompatibility of that particular biomaterial. So that this is how you can understand how our body responds when a biomaterial is present or is introduced. So I tried to summarize everything. It was discussed few lectures, but in very short I tried to summarize. I might have gone a bit fast for some of you. So if there is any query, you can ask. Any queries? Have you understood, all of you? Yes, no? Okay. I hope others have also understood. Okay. So I got thumbs up from two people only. All right. So if you have any query, we have time, so you guys can ask later on as well. So there are various in vitro tests that we do using cell lines onto the biomaterial in order to answer whether it is going to be biocompatible or not. Okay. So there are some qualitative assays and some quantitative assays, and there are some hemocompatibilities assays as well. So qualitative assays will like help you answer whether it's yes or no kind of a thing, whether it is working good or not. Quantitative assay will help you like tell you with surety with using numbers. All right. So in vitro test, we have to learn cell culture practices, all right, and under sterile conditions, we grow cells on top of any biomaterial in question and see how the cells adhere to it, how their morphology changes, how well they are proliferating. So, and if it is if it is supposed to differentiate, then into what type of cell it is differentiating to. Okay, then if the biomaterial is causing cell death, then what type of cell death is happening? Is it happening due to necrosis, or is it happening due to apoptosis? So and so there are various kind of questions that gets answered using this kind of in vitro test. So some of the qualitative assays that one can do is check the cellular morphology, check how the cells, whether the cells have adhered or not, and the cell number if it is increasing in time. And these all things you can do using microscopy, either in presence of some dyes, all right, or you can use some bright field microscopy where no dye or anything is required to observe the cells. So simple microscopy, or using any compound light microscope or a fluorescence microscope, okay? Or if you are using a 3D substrate, then a confocal microscope can help you to answer these simple questions. Now, if your biomaterial is uh, is not supposed to cause cell death, but if it is causing cell death, then you can see it. You can actually distinguish between live and dead cells using microscopy, where you use specialized type of dyes. One dye is called propydium iodide that specifically help you understand which of them are dead cells, and another kind of dye is called fluorescein diacetate. Then there are other type of dyes as well. So we use in our lab, we use fluorescein diacetate, so that permeates very well within the live cells, okay, and gives a green fluorescence. So for this type of assay, you need a simple fluorescence microscope to understand how much of the cells are dead, how much of the cells are alive, and then it will help you understand whether the biomaterial is causing cell death. Now, there are various pathways for cell death. You can answer whether it is going through apoptosis. So, apoptosis, if you have any idea, so apoptosis is a mechanism where it is the cell fragments, okay, breaks down into smaller fragments. So, that is why it is a very silent form of death. It doesn't cause any um, mess in the tissue area, okay, and the cell membrane and all, they all stay intact kind of, and only the cell fragments, the DNA material, everything, it becomes fragmented. It becomes smaller first, it becomes condensed, and then gets fragmented into smaller vesicles kind of structures, which are called apoptotic bodies. So if the cell is undergoing apoptosis, we can understand that by using some dyes, some fluorescent dyes like acridine orange, or using another dye that is called ethidium bromide. Okay, so using these dyes, we can stain the cells and see if these kind of apoptotic bodies are present or not. Then, in order to understand whether your biomaterial substrate is able to provide a good substance for cell migration, for that we do scratch test, where using something sharp, we just like spontaneously make a scratch, all right, and then we observe how uh, how much of the scratch area is being covered with cells over a period of time. So we don't add cells afterwards. Okay, we on a monolayer of cells, we just make a scratch. We're using anything sharp, using anything pointy, and then observe whether that particular area is get covered with cells or not over a period of time. So this helps us understand that the area got covered because the cells proliferated and migrated. So that means it's the cells are healthy and they are growing very well on the biomaterial. Then there are other methods as well, like extract dilution. So if the biomaterial is supposed to degrade, or if during polymerization some um, monomer of egg is like remaining behind, then you can see whether that particular monomer component is cytotoxic or not, whether it is causing cell death or not. So you basically grow cells in presence of a polymer, or like solution, basically some extract of your biomaterial. Then direct contact, you just grow your cells on top of biomaterial. Okay, you grow a monolayer if it is growing very well, and then you observe the cell morphology, etc. And if it is, of all things are good, like cells are growing well, it is adhering, it is proliferating in this case, then your material is cytocompatible. Then people also do agar diffusion. So this is another way of testing whether the cell is able to migrate and then adhere to the surface. So in these two assays, here we just see the effect of the biomaterial on the cell morphology, cell adhesion, cell proliferation. But in agar diffusion test, we also see whether the cells are able to migrate. First, they have to migrate. So agar is basically creates a layer of separation between the biomaterial and the cells. So in order, so if the cell is able to respond if to the biomaterial, or if it has to adhere to the biomaterial, it has to migrate first. Okay, once it able to migrate, then only it can adhere to it, and then it will be able to gain its morphology, its required morphology, and proliferate and cover the entire surface of the biomaterial. So these are some qualitative assays, and it helps us answer very basic questions, okay, about whether the material is cytocompatible or not. Then we can quantify this question, we can give quantitative answers to this question using some quantitative assays. So first is called MTT assay. So here we can measure how much of the cell is viable. Okay, or you can also say how much of the cell is remaining viable over a period of time. This is a very powerful essay, and it can help you answer various important questions using numbers, and people value numbers. Similarly, there is MTS assay, which is very similar to MTT assay. The only difference between these two assays is the type of the salt that we use, and that only remains different. Other than that, the steps, the basic steps and all remain same. Then there is LDH assay. So this particular assay is based on the fact that if the membrane of the cell is compromised, then whatever thing is there, enzymes, etc., it will come out. So along with it, an important enzyme called lactate dehydrogenase, that enzyme will also come out. So if the cell membrane is compromised, so that is the first step of cell necrosis basically. That means you can say that the cell is dying through the necrotic pathway. So which is why we use this particular assay to quantify whether cell necrosis is happening or not. We can actually do a time dependent study as well, where we can quantify the level of LDH, okay, over a period of time, and see how toxic our biomaterial is. Okay, then if your material is going to come in contact with blood when inserted inside the body, then we have to also see how it is impacting your blood cells. So first test that we do is hemolysis. So the biomaterial can, if the biomaterial causes damage to the RBCs in vivo, so that question can be answered in vitro level as well. So you take a vial of blood of any patient, but you have to make sure that it is a normal patient, a healthy patient who is not under any type of drug for any kind of heart disease or blood conditions. Okay, it's a normal healthy patient, and from that person you have to take a vial of blood and detect, and you expose his blood or her blood to your biomaterial and see over a period of time how the RBCs are changing its morphology. Okay, then there is coagulation assay. So here it is a time dependent kind of an assay where we see whether our biomaterial is able to cause coagulation, and if yes, how early it is or how delayed it is. Okay. Then there is complement activation assay. So here again we see if the biomaterial is able to cause any changes to blood plasma, and in return the complement factors which are there in the blood plasma, whether they are getting activated or not. So these type of hemocompatibility assays can give you some idea on how your material will behave when it is inside your body and in contact with blood. So I hope you have understood a brief summary of all the in vitro assays that one can do to test whether their biomaterial is cytocompatible or it is cytotoxic. So based on this understanding, I have few questions. So this is your first question. Is it visible on your screen? So the question is, which one in this list is not an in vivo test? So in vivo means you use an animal model, and in vitro means you use cell lines, okay, to whatever, to answer whatever question that you're interested in. So these are the four tests given in the option. First option is pharmacokinetics, second option is MTT, third option is pharmacodynamics, and fourth option is histopathology. So which of these four tests is not an in vivo test? Or in other words, I can say which of these in this list is an in vitro test. This is another way of framing the same question. Anyone?
MTT.
Yes, you're correct. The correct answer is MTTSM. So all the three types of tests, they are done in vitro. They can only be done they can only be done in vivo. I'm sorry. So MTT is the test that you can do at in vitro level using cell lines. Now, a brief summary on what types of in vivo test that we have to do in order to um, basically convince the regulators to use this, to approve the use of the of your biomaterial for human use. So there are levels, okay? So when you have, like, suppose designed a novel biomaterial, you first test it using cell lines, okay? Where where you don't need basically any type of approval from any particular body. So you just have to order cells and appropriate media and test it. Once you have the results of in vitro level testing, then you move on to in vivo testing. Now here, the job done in vivo is actually much more difficult because you're uh, dealing with live animals, all right? And using those live animals, you have to answer few of these questions which I've shown in the paper, in this slide. And then once you have those answers, you can then further go on. So this in vivo test is comes also under the preclinical trials, if you have heard it, okay? And then it goes to maybe the phases of human trials, the phase one, phase two, phase three. So after the results of this in vivo test and in vitro test can help you convince the regulators, okay, to pass your biomaterial for human use. Together, you have to show the data. Now, in vivo test, we answered many questions. So first test is sensitization test and irritation test. So here one tries to answer whether the biomaterial is able to cause any local or systemic allergy or hypersensitivity reaction when it is exposed, or when the person, or whether when the organism is exposed to it for a long period of time. So when I'm talking about allergy or hypersensitivity, here what happens, um, that any component of your biomaterial, it can cause unnecessary autoimmune kind of reactions, okay? Or it can trigger the release of cytokines, and it can cause superficial symptoms like rashes, like anything, nose bleeding and all. Like these are all hypersensitivity reaction, means your body is not meant to have that particular material into your body. So so if you have heard the allergen terms, like many of the people have allergies towards cutaneous fishes or towards mushrooms. Similarly, people have to test if similar kind of tendency is there towards their biomaterial. Then there is acute and subacute systemic toxicity testing. So acute means for a short period of time, and subacute, so acute if you're saying for maybe 24 hours, 48 hours, then subacute is like longer than that, okay, 1 month maybe. So you have to test whether the biomaterial is causing overall body toxicity, means systemic means throughout the body. The first test was like local, at the site of implant, as well as systemic throughout the body. The second test, you have to test throughout the body if what kind of toxicity is seen when tested for a short period of time. And this is done using very small animals like rat, mice, rabbits. You don't need large animals to answer this question. Then there is subchronic systemic test. So this here you test whether the material is toxic for a much longer period of time, maybe years. Okay. Then genotoxicity. Here people try to understand whether your biomaterial, the component of the biomaterial, or degraded byproducts of the biomaterial is causing some gene damage. Means it is causing some point mutation or any other kind of the mutation, which indirectly causing alteration of a particular type of protein or causing alteration in particular type of metabolism. So these kind of tests is also done. Then implantation test, it basically tries to see whether the material is able to do its job when implanted inside the body. So for complex type of biomaterials like say pacemakers, okay, or heart valves, where you have to introduce some electric component, all right? So and then it becomes a challenge for the maker who have made this to answer that it will work perfectly inside the body as well, okay, because various complex factors come into picture. So that is why implantation test he has to do to prove that this material works very well inside the body. Then hemocompatibility test, just like you did in vitro, you have to also do in vivo because the conditions have become slightly different, and like the number of cells, the compositions of the blood, so all these things also have plays a very important role. So hemocompatibility test you also have to do in vivo. Then carcinogenesis bioassay basically tries to understand whether the biomaterial is able to cause or has the potential to cause cancer or make healthy cells cancerous, change its phenotype to a cancerous type. So people also test it. Then whether the biomaterial is safe for use for pregnant women, say, okay, because they are carrying his or future generation. So in that case, you have to test whether the material is able to cause any defect to the growing baby, okay, any developmental defect. Or even in men also, people have to test whether it is causing any damage to to his reproductive organs, okay, so that means the reproduction function, it should not be hampered due to the use of the biomaterial. So that toxicity level, if it is happening or not, if it is damaging, then it cannot go forward. Then there is pharmacokinetics. So here it makes sense when your material is biodegradable. Otherwise pharmacokinetic studies are done for drugs, not for biomaterials in general. But if your material is biodegradable, it is supposed to degrade out, removed from the body. Then you have to study how fast it is getting removed. You first insert it somewhere or you eat it if it is oral, okay, and then it will be absorbed, okay, and then excreted. So there are steps: first distributed, absorbed, then excreted. So these are the steps of pharmacokinetics. So this similar type of thing has been also been made, or steps also been made for biodegradable materials. That is why pharmacokinetics you need to study. Then safety trials. So you have to do additional test to answer two important questions that any regulatory authority ask for a drug, even for a biomaterial, whether it is safe to use and how what is the efficacy of your biomaterial. Does it work every time? And if does it work every time safely? So this particular question has to be tested, or have to be answered using appropriate results. Then lastly, people also do histopathology, where they check if your biomaterial has caused any damage to the tissue or the organ structure. So the major organs of our body, like heart, liver, kidney, brain, then all the major organs. Okay, so they spleen, stomach, and lungs. So they do sectioning of. They take small sample of each of these organs. They create tissue sections out of it, and they use different kind of stains, tissue stains, and observe whether the tissue structure is altered or not in presence of your biomaterial. So these are some in vivo tests that people has to do in order to move forward and answer to the regulatory authorities whether the biomaterial is safe, and whether it is able to perform efficiently inside the body, and then it can move to human trials, the phases of human trials. Now, I have another question, which is similar to what we have discussed so far, but it is majorly based on the concept in the lectures that Professor Doble has introduced. So here there are three columns in this question. The first column is the type. This is contains the name of the biomaterial. Second column tells you the contact duration of the biomaterial, uh, and the third column tells you the list of or the combination of in vivo test that you have to perform in order to approve it. So these are the three columns. You have to basically match uh, the options with each other. So we'll go one by one, okay? One biometer at a time. Acha, have you guys understood the in vivo test, or what are the tests that is performed in animals? Have you guys understood that? Okay. So now moving on to this question. So first biomaterial is a nicotine patch. Does all of you know what is a nicotine patch? How does it work? I'm sure you guys have heard this nicotine patch. Anyone would like to explain to others what is a nicotine patch? No one? You guys are not aware? So basically, nicotine patch is a transdermal drug delivery kind of system. So here, drug is the nicotine, and it helps the user to, over the period of time, stop using cigarettes. Okay, it helps in that. So nicotine patch, they are applied on the skin, and they deliver the nicotine level slowly to blood through the skin. So they are, that is why they are called transdermal patches. Now tell me, since they're applied on skin, and then what should in column B there are three options. How would you categorize how long should a nicotine patch be used by a person? Should it be less than 24 hours or 24 hours? Should it be more than 24 hours, like 24 to 1 month? Or should it be used permanently? What do you think? Any idea?
Later 24 to 1 month.
Okay. So here in nicotine patches are actually used one patch, so it comes as a packet. So one patch a person can use for 24 hours, then he has to remove it and then use another. And a doctor recommends that the usage of this patch should be continued for 8 to 12 weeks, not more than that. So one patch, the life of one patch is 24 hours. So for 24 hours a patch has to do its work, which is why one matches with option B. The contact duration of a nicotine patch is 24 hours. Now tell me, This is the specification for with which this particular biomaterial is designed. Now tell me which combination of in vivo test you have to perform to approve its use. A, B, or C? Means you have designed a nicotine patch and that will be used by the user for 24 hours. Now you have to do the test so that it can be approved by the regulatory authorities. So what test will you do for this particular biomaterial?
Ma'am, option B.
Option B. So why do you think it should be option B?
Ma'am, because it should pass the sensation test so that it does not cause any irritation to the skin. Secondly, as it is acute systematic toxicity, that how much time it would take to, toxify that,
So
and
does
it will not have
yes.
any implantation effect on the persons, and also genotoxicity. I don't think that it would affect. So that's why I chose option B.
Okay. So I have another question in the same line. Do you think this nicotine patch will come in contact with blood directly?
Not directly, but indirectly, it may.
So the nicotine is the compound that is diffused through the skin and reaches your blood vessels or blood, right? So the material that has been used to create that patch is the material in question in our case. So according to me, we don't have to worry about blood contact. So hemocompatibility test is not needed. And coming to systemic toxicity, so you are just keeping it for 1 day for 24 hours. Toxicity can arrive when you are like going to keep it for more than 24 hours, like the use is prolonged. Then systemic toxicity comes into picture, means because the material needs that much time to cause toxicity, our body is very much resistant to normal uh, anything, but we make it susceptible by adding something foreign to it. And this particular, in this example, nicotine patch, it is just on the top of the barrier, right? Skin is a barrier function. The barrier is going to anyway help us to uh, keep all the foreign things which can cause toxicity away. Got it? So systemic toxicity is not required, but we have to see, since it is on the top of your skin, so it can cause some rashes, it can cause irritation, it can cause allergic reaction, right? Then it can be toxic to our skin cells. So which is why the correct option is C here. Okay, we just have to test three things. And why? Because first, the contact duration is very small, only for 24 hours. And second thing, it is not coming in contact with the blood. All right. And for causing acute systemic toxicity, you need a longer period of time. So the most appropriate option is C. So everyone got the logic here, what I tried to explain, everyone got it? Okay. Now, similarly, we have to think over for the other two options. Second option is the feeding tube. So you guys know what is a feeding tube? Anyone heard of it? So feeding tubes are used for those patients who cannot eat on their own, means they cannot chew the food and digest it. So the doctors have to insert a tube passing through the gastrointestinal tract and it reaches the stomach. And through this tube, food is delivered to the stomach. Okay. Now, tell me. So this particular tube, it doesn't directly again come in contact with blood, but since it is inserted within our gastrointestinal tract, so there are blood vessels there. So that in that case, it can easily rupture those blood vessels. So it is indirect contact with the blood. Okay, it is blood doesn't exactly touches the surface if inserted correctly, but if it is not, then it can definitely damage those blood vessels and blood can come in contact. So this is how the feeding tube works, and this is the contact surface. Now tell me, what should be or what do you think is the ideal contact duration? Like for how long? So there are only A and C. Okay, so you have to choose between these two options. So what is the contact duration for a feeding tube? What do you think? Maybe a guess.
Less than 30 days.
It is greater than. Are you referring to option number A? It is greater than 30 days.
Greater than 30 days. No, it would be C. 24 to 30.
Yes, 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 you're correct. It should be C. We don't need it. Usually doctors don't use feeding tube. The normal way is to eat on their own. They do it on special situations only, right? So you're correct. Now tell me, in option among option A and B on the third column, which type of in vivo test should one perform on this feeding tube?
B.
Yes, you're correct. Now you guys got it. All right. So I think it is understood now. So the last option remains is the left ventricular assist device. So what is, I don't know if you have heard it or not. So what it does, it helps, it is inserted in those patients whose hearts are not functioning and they are in need of heart transplant. So these assist device acts like a temporary heart and until the patient receives a heart transplant, okay, transplantation. Sometimes it is like it just helps the person to live for a few more years until heart is made available, but many a times heart is a very difficult organ to find a replacement for, so the patient dies. Okay, so you can say in this case this device is meant for the lifetime of the patient sometimes in some scenarios. Now obviously option A is remaining, so this particular type of device has to be tested or will be in contact in the body for a very long period of time. So which is why the contact duration is more than 30 days means it is like a permanent kind of a thing. And since it is permanent, we have to be very careful about, and we have to test all the tests that we saw in the previous slide, almost all of them. We have to even test the genotoxicity here if the material is causing some type of point mutation or any other type of mutation and causing again some toxicity level. So I hope this question is clear to you all now. So this is the answer given. Now tell me, uh, based on what you have understood from the lectures actually, the artificial heart valves. So people are making artificial heart valves as well. So if you have made it, then in vivo, which kind of animal model would you use to test it? Would you simply just think, would you use a smaller animal or a bigger animal to actually check whether this material will work or not? The options are rat, you can choose a dog, sheep, or a rabbit. Which animal would you use?
C.
Sheep, you're correct. This is directly from the lectures, okay? That is why I'm not going very deep in here. Then the treatments for diseases that affect joints. So these treatments has to be tested in vivo. But on which animal? Would you test it on rat, goat, sheep, or rabbit? Anyone? It is goat, right? So you can go and refer to the lectures. Okay, there you will find it. So goat is a ideal model actually to study the muscle and the bone joints and all. Then, which of the following factors should be considered while selection of animal models for research? So, you are a researcher and you have made some biomaterial, and you have to now justify its use in human. So, to do that, you have to first test it on some animal model. Now, you don't know which animal model to choose. Okay, you have to do a selection. You are given all your, you have enough money and everything, and you have your own animal house, and you have the option of choosing to do experiment with any of them. But how would you justify your selection of animals for your experiment, or what factors are important here to consider when you are going to choose an animal for your experiment? So there are four options here: appropriateness as an analog to mimic the real life condition, cost and availability, generalizability of the results, all of the above. This is again directly from the lectures. This question is directly from that. Anyone?
All of the above.
Yes, you're correct. So it's nothing very complicated here. Just go back to the lecture. It is there, like sir has given in bullet points what factors to consider. Okay, that is why I'm not going in very detail. Now, so o- any so you are a researcher, you have decided to test your particular biomaterial on an animal. So any committee will be there to basically question you, ask you several questions, and you have to be uh, answerable to them, because after all you are going to kill a life for research. That's a very, very big thing. And there are special committees, ethics committees are there at institution level, at country level. So these committees are there to make decisions, okay, whether this experiment should be done or not, whether the results of this experiment is going to be of value to society or not. So they are there to judge. You have to go and make your case, and then your particular idea or a research proposal will get accepted. So when you are trying to design or plan such an experiment on animals, you have to make sure to follow 3 R's. So what are those 3 R's? First is you have to question if I can not use the animal, means if I can test my hypothesis on an alternative model. Okay, so I have, so if, is there a replacement, is there an alternative to not using animals? So that is your first principle that you should have in mind, because the same thing will be asked by the committee as well. Like, have you done any research that this should be done on animals only? Can it be done on any other models, or can you prove your hypothesis in any other way? Okay, so that is the first question. The second question is reduction. So you have to plan your experiment in such a way that you are going to use less number of animals, all right, and get the data. You don't have to kill so many animals, because most of the experiments will ultimately lead to sacrificing those animals and then testing their tissue structure and etc. So you have to start thinking that only this very like minimum number of animals I should start with. I shouldn't have too many replicates of my experiment. Okay. And their committee will also ask, do you need this much animal? Don't you think it is too large? Okay, if they ask you like this, then you have to justify. Ki, no, I need this much only to validate my results, and this many replicates I need anyway. Okay, so you have to justify this. Reduction is another R. The last is refinement. So refinement means, if possible, we should be able to plan our experiments in such a way that if animal does not have to be sacrificed, if it is possible, or if it is have to be sacrificed, then the method of like whatever method that you are using, it should be less painful. Okay. Or if you are rather than using replicates in your experiment, you can try to use a biomaterial and implant it at different locations within the animal, so that you get more data out of one animal. So in this way, you should refine your experimental plan such that there is less animal suffering and you get maximum amount of data, and that will justify killing these many animals. So these are the three R's that a researcher who is working with in vivo models or animals should keep in mind. And these questions are also asked by the committee as well, who are uh, there to going to pass, who are there and will pass the proposal. Okay. Now, we have briefly covered the in vivo, in vitro, and the biological response part of the course. Now, I'll briefly discuss few problems on material. Okay. In the next class, we will go much more deeper into metallic biomaterials, polymers, etc. Now, this is a question in front of you. You have to answer silica glass. Silica glass is a type of biomaterial. Now it belongs to which category of biomaterial? Is it organic material? Organic means generally nature derived. Then is it a metallic material, a polymer, or a ceramic? So, how would you categorize a particular biomaterial? And like how would you decide that this particular biomaterial should belong to metal or should belong to polymer? Anyone has any idea? Or they has just been told that this particular material is polymer, that's it. Have you ever questioned yourself why it is a polymer, or what characteristics does it have, so that we call it a polymer? Something like that. Has any one of you given a thought? Or another way I'm asking, if silica glass here is given to you, and as a biomedical scientist, you are told, uh, now put them in a bin named labeled as metal, polymer, ceramic, or organic. So in which bin will you put the silica glass? You should if you are able to answer, that is good. But I would also like to know, how did you answer it?
Polymer, ma'am.
Okay. Okay, so I've received one answer. I will not tell whether it is right or wrong. Anyone else would like to try? It's okay if you're wrong. You're just here to learn. So there should not be any hesitation. Just give it a try.
Ma'am, a polymer.
Okay, so there are two people who are in support of polymer. Anyone else has any other question? Any other answer to give for this question? Okay. Now I'll give you the answer, but first I'll show you how I came to that answer. Okay. So what is silica glass? Silica glass is basically composed of silicon. Silicon is the most abundant element on the earth. Okay. And it is bonded, covalently bonded with oxygen. So if you were to ask the unit of silica glass is basically SiO2. It is amorphous in nature, means the arrangement of silicon ions and oxygen ions such that it is unordered. All right, it is amorphous. It breaks very easily. So it is brittle as well. Okay. Now, silicon glass is the unit, one unit, and similarly, it can bond to like various silicon glass like this. So silicon oxide. So this is called silicon oxide. Silicon oxides are basically these molecules are basically covalently bonded like this. So in this way, a lattice, amorphous type of lattice structure gets formed. So in this particular material, there is no carbon, no hydrogen, okay, no nitrogen, no phosphate, nothing. Oxygen is there. So if you use your previous knowledge, your class 12 science knowledge, then you know organic material which is of nature origin usually have these particular elements in it, which is why it is not a organic material. Okay. Now, when you look at the structure of silica glass, the bonds are mainly covalent, which means there is sharing of electrons. And when you look at the metal structure, there is something called the metals, there's something called a metallic bond is present, where the positively charged ion is surrounded by negatively charged electrons. And these electrons are basically holding this positively charged ions like a glue. Okay, this is a metallic bond kind of a structure. So this kind of thing is not there, as I've explained, right, in silica glass. So it is not a metal as well. Now, what are polymers? If you know the basic definition of polymer, polymer is made up of monomers. Okay, and these monomers they are repeated n times. So does silica has a similar structure like polymer now? Anyone? Yes, no? Those who have answered polymer, you guys should now answer.
And the slide is in still 3 hours. Ma'am.
Yeah,
Slide here, still 3 hours.
I'm not getting. Uh, 3 hours means what?
The slide
You,
has changed it, ma'am.
yeah, you're not able to see the slide.
Yes, ma'am.
Okay, so let me reshare my screen. Okay, I have stopped sharing. I think the screen you are not able to see now.
Yes, ma'am.
Okay, can you see the slide now?
No, ma'am. I guess my tower is weak. Continue.
Okay. What about others? Are you able to see the slides? Okay. So I received a thumbs up from one of you. Okay. So as I was saying, silica does not have a polymer-like structure. Like there are no monomers connected with each other, right? And it is not a chain-like structure. So it is not a polymer. The remaining uh, option is ceramic. So if you look at the characteristic of silica glass, it is brittle, it is amorphous in nature, all right. And there, in ceramics, there sometimes ionic bonds are also present, but covalent bonds are also there. So both covalent and ionic bonds usually are present in ceramic type of materials. So here only covalent is there, but that will work. So it is a ceramic material, which is why it is a ceramic material. So you have to reason like this, why this material should belong to this particular class. So maybe some background knowledge about various type of materials and how their structure looks like can help you understand and answer these type of questions. Now, hydroxyapatite. You guys know what is it? It is one of the major component of our bone. So the mineralization of the bone is because of the deposition of hydroxyapatite. So a hydroxyapatite is what? There it is the question. What is hydroxyapatite? The name sounds very fancy, but if you look at the chemical formula of hydroxyapatite, what it is. So to answer this question, you have to first uh, maybe Google, right? What is hydroxyapatite's formula? So if you Google, I just did. So the formula comes out to be Ca10 PO4. This is 6OH2. This is the formula of hydroxyapatite. Now tell me, which of these four options seems correct to you? Is it a hydroxyproline, calcium carbonate, calcium phosphate, or calcium sulfate? So hydroxyapatite, this is the formula of hydroxyapatite, which is there. Are you able to see my screen? I have written it on the slide. So it has Ca10PO46OH2. So what is a hydroxyapatite? Is it a so it is not hydroxyproline. It is clear from the formula. So but it is it a calcium carbonate, calcium phosphate, or calcium sulfate? Very easy question. Just you have to yeah, someone has unmuted themselves. Would you like to answer? ओके फिर से म्यूट हो गए हाँ बताइए
Calcium phosphate.
yes you're correct it is calcium phosphate great now which of these is a dental biomaterial means biomaterial used for dental applications so there is titanium or type of titanium so Ti starts for titanium then hydroxyapatite amalgam or stainless steel SS is 316L low carbon stainless steel so various metals, um, ceramics are used for both dental applications as well as for orthopedic applications. But the requirement of strength is different for teeth and bone. So based on that judgment, and what you already know about titanium, hydroxyapatite, and stainless steel and amalgam, tell me which of these material can be used for dental application. Anyone? So we actually saw the
Hydroxyapatite.
hydroxyapatite. Okay. So actually it is not correct. It is amalgam. So if you look at the Young's modulus of each of these materials, so that is already been shared, if you remember, in the lecture slides given by Professor Doble. So there you can see the Young's modulus of each of this material. So among these four material, stainless steel is the strongest material kind of, whereas and titanium are nearby. Whereas amalgam is the most weak material, is among four of them. And teeth is not as strong as your bones, right? So amalgam, that is why, is used for fill cavities and all, and as well as to support your decayed tooth. So amalgam here is the correct answer. Maybe go back to the previous lectures like week 1, week 2. So there the introduction to biomaterial was given. So there these materials were discussed. Okay. And also the next week I'll definitely discuss the metallic biomaterials part. So then also you can ask question if you have on this particular topic. Now, which material has a very high elastic modulus? So for answering this question, if you have your computer, phone, laptop, whatever device you have joined, if you like to open assignment week 2 assignment, if you open, okay, and see question number 1, where there is stress versus strain graph, and there are four plots are there. A, which looks like this, B, C looks like this, and there is D. So I've replicated that particular plot in my slide as well. Now, if I ask you, this is the question. So first question, before you come to the main question, first question is, which material among A, B, C, D has a very high elastic modulus? From the graph, you tell me. You guys know the definition of elastic modulus, correct? Which has already been taught. Now tell me, which among these plots, A, B, C, D, has the highest elastic modulus? One by one we'll go. Anyone? So look at the uh, slopes of this elastic region. Elastic region is the linear region. Look at the slopes. If my graph is not that pretty, you can go and refer to assignment 2, week 2 assignment 2. And there you see the linear region first. Okay. And see how long it is. So if you observe carefully, for graph A, the linear region is the longest. Okay. And it is not transforming to curved region that well. The curved region, that is the plastic deformation region, is comparatively very less. So, and if you compare so what is slope? Slope is basically how sharp the line is. Means sharp means how inclined, you can say how inclined that line is. So if you see graph A, the line is pretty straight. Means if you increase the stress, the strain increase. The strain will not increase to that much magnitude. It can take a very sharp increase in stress, but the proportional increase in strain will be very small, right? So, which is why of graph A has a sharp slope, very high value of slope, and it has a and slope of stress versus strain graph is what? It is elastic modulus. The value of elastic modulus for graph A is high. Now, next question comes. Okay, A has very high elastic modulus, but what is this material? Okay, what is this material? Like, which class of material does it belong to? Is it a polymer, metal, ceramic, or a blend? So, and blend is like a very arbitrary term. A blend can be of anything, a polymer ceramic blend, polymer polymer blend, polymer metal blend. So, blend is not the option here. Okay, you have to think whether A is polymer, metal, or ceramic. Anyone? So, you have already done this question. You've already done this question in assignment 2, and you know what is A, right? You already know what is A. It is already evaluated. The A was hydroxyapatite cement. And if you know which class of hydroxyapatite does it belong to, then your answer is done. Have you understood the logic of how to answer this question? If yes, go ahead and give it a try. Don't confuse the numbering of the options with A, B, C, D in the graph. Okay, don't confuse. It is different A, B, C, D, and the labeling here given in the option is different. Focus on the names. So what is A here? Because A plot has the highest elastic modulus. Which material can have now elastic modulus very high? You already know A belongs to hydroxyapatite. So what is hydroxyapatite? Is it a ceramic, a metal, or a polymer? Which class of biomaterial does hydroxyapatite belong to?
Ceramics.
Yes, you are correct. Ceramics is the correct answer. So, Santosh, can you tell everyone how did you arrive to this answer?
Uh, Ceramics are stronger one, ma'am,
Okay,
like
but they,
hydroxyapatite.
yes, but they're brittle as well. So
Ah,
their plastic region is not that long. Okay, it will fracture, like it will break very easily.
wait.
If this, yes, correct. And so in this graph, A is a characteristic graph of a ceramic material or in this case, hydroxyapatite, and hydroxyapatite belongs to ceramic. So now others have also understood, those who all are like listening, not answering. Have you guys understood? I got one thumbs up. I should have
Yes.
received three thumbs up. Okay, okay, great. Now, a very simple question. I think this is the most easiest question that we have discussed today. So, which of this category of material does have which have very high electrical conductivity? Very easy. Like you have been studying this for maybe like 1, 2, or maybe
Weak
5,
metals.
6. Yes, correct. Now, can anyone use your knowledge of biometals, like whatever you have studied, to answer why metals are highly conductive, electrical conductive? Now, that is a important question, and that we should know at this level. We are just taught that metals are highly electrical conductive, which is why we have metal wires to carry current in our and metal um, like elements. But why metals have high electrical conductivity? Anyone would like to give it a try?
Less intermolecular space.
Yes, yes. Can you again repeat?
Less intermolecular space.
Okay. So you are kind of correct. Okay, partially correct. But so the metals, as I told you, there exist metallic bonds. So the metallic atom here is positively charged. It is a cation itself. It doesn't have it doesn't need that much electrons. It is electron donor, you can say. So it makes itself positively charged. So these are the cores, you can say. And these these cores, positive cores, are glued together by electrons. These electrons are free, kind of, not completely free. They just they can move up to certain distance because of some space that exists, but they are kind of free. Okay, more freer than in case of ionic bonds or covalent bonds. So which is why when electric current comes, these electrons are free to carry that so this electron becomes they can travel easily, which is why they are electrically conductive. Similarly, the electrons help in conducting heat as well. These the metals are very good thermal conductors as well. So this the same logic does not apply for polymer ceramics because their molecular structure, atomic structure is completely different. Their different bonds are there. So you cannot use this particular logic for them to answer. But for metals, this is the reason. Now, like, I don't have much time today. I was thinking maybe I'll finish 30 minutes before, but I was thinking to discuss few of the problems or all of the problems of week 4 assignment, because I saw in the course portal that many of the students have some difficulty. So have like 10 minutes or so, and in this 10 minutes, if you guys faced any difficulty in understanding any of the problems in week 4, that we can discuss. Okay, and rest of the problem, I think uh, you have done it already, so I think you can do it. But anyway, I'll slide this. I'll share this slide, and in this slide you can later on refer to the problem, a solution of week 4 problem. But if you personally have faced any difficulty in understanding any of the questions of week 4, or your particular answer was not correct and you don't understand why, so maybe you guys that is the right time to ask. Maybe I can help you a bit. Okay, so let me go one by one. So if you have your assignment 4 open in your uh, device, so first question is asking about which process occurring in biofilm system has the highest time scale. By highest time scale, it means to say which process takes the longest time when a biofilm is kind of forming. So the first process is convection. Convection is nothing but uh, motion of the fluid and the motion of the bacterial cells within the fluid is referring to that. Then there's viscous dissipation, then substrate diffusion, then biomass growth. So the correct answer for this question is biomass growth, because above three options will help the bacterial cell to kind of reach the surface of the biomaterial. Now, once and these processes itself takes very small time. It is dependent on other factors like how viscous the media is, or how light or less viscous the media is, then diffusion rate it depends on. So those happens at a very small time scale. It happens very quickly. Whereas when it comes to proliferation of bacterial cells, say let's say E. coli, uh, doubling time is around 12 hours or 24 hours. That will take time. So among these four options, biomass is the correct answer. Now option number two, So here you have to calculate what is the length of the longest diagonal. And the correct answer is in the range of 0.5, sorry, 5.5 to 6.5. So I have already discussed this in my last week problem solving session. But if you have not been able to answer it correctly, let me know. Has everyone, like who all are present here, have you all answered it correctly? Is your answer correct in the portal?
Yes, ma'am.
Yes. So yeah, so that's why I'm skipping it. Anyways, you know the logic then. You have done it, then you know the logic. Then the next question is based on the Bragg's law. So here, in the screen in front of you, means in the slide, I have shown the equation of Bragg's law. And if you just see the definition of each of these variables and the constants in the equation, from that definition, you will be able to answer. So the correct answer here is option B. Then for question number 4, so the correct answer is 0.16, by the way. All right. And if you have answered 0.16, then it will be rectified. This assignment will be rectified, and so the typical range of accepted answer is given here right now is not correct. The correct answer is 0.16. I can show you how 0.16 came, but first tell me what answer did you get while solving question number 4?
0.16 only, ma'am.
0.16, no? Okay. So others also, only one person is answering. So I'm assuming others also did the same way, or is same for others as well. I'm assuming that others also got 0.16.
Yes, ma'am.
Okay, great. So that will be rectified. Okay. Now next question is based on FTIR analysis. So the correct answer here is option number C, 1600 to 1700. So for solving this question, you had to go and check the chart, FTIR analysis, data analysis chart. The chart which gives you or tells you for which type of bond you are going to see a peak, right, in your FTIR plot. So that chart is helpful here. And in your slide, I have done a job myself. So I've referred to a book, a very famous book of spectroscopy by Pavia. All right. And from that chart, I got to know that in the range of 1680 to 1600, alkene bonds exist. Then 1680 to 1630, this type of amide bond exists. All right. Then 1600 to 1475, aromatic alkene exists. Now, in the options, if you go, so they are asking for the group. Okay, now this they are not saying which group directly, by the way. They are saying ki your group belongs to a protein, and you know your protein or the unit of protein is amino acid, and amino acid have major groups like carboxyl group, then NH group is also there, NH2 group, carboxylic acid group. So these groups are main component of any amino acid. Then R is also there. Then R is like anything else, and based on that, the classification of amino acid is there. That is not the point. My point is you have to find that particular functional group of a protein in your chart, correlation chart, and see which for and see what where would you find a peak in your FTIR plot, and in what is the range of wave number where you can expect a peak for that particular protein group. So first option was 1500 to 1600. Now based on the values that I've shown here, this is actually this particular option 1600 to 1475 matches with option A. But usually proteins, if you see the backbone of amino acid, it does not have aromatic alkene there. Maybe R group can have. Okay, if you check, this is how the protein looks like, the basic structure of amino acid. R can be anything. We are not focused on R now. Okay, so so A is not the correct answer. Then 3000, 3300. Again, you have to go to the chart and you will see that option number. So this particular group, alkenes, CH group of alkenes is actually under option number B, 3000, 3300. So here I'm getting a peak between 3000 to 3100. So this is option number B. But in the backbone of amino acid, alkene CH is not present. So it is not correct. Then go to option number C, which is the correct option here. But while solving, you didn't knew 1600, 1700. Again, you have to go back to your book and check what is the range matching and what is that functional group. So you see uh, for 1630 to 1680. Now, although it is not exactly the same, note it is not exactly the same, but the range that is given in your question and the range that you're finding in the book, it is fitting. Okay, it is within that. And this particular range of wave number is where you will find the peak for amide. Now, if you look closely, this is a type of functional group which you can expect in an amino acid, because here there is N, right? And when this one amino acid is bonded to another amino acid through a peptide bond, remember peptide bond? So there you will find C double bond O, and there will be NH as well. This kind of peptide bond exists. So option number C is that's why correct. So if you have done the logic or you have used the logic in this way, you would have arrived to this answer. Is option number C is correct. And option number D is like this. But again, I couldn't find any functional group which gives a peak between 680 to 860. So this is the closest I could come. But again, this particular CH aromatic is not found in amino acid or in a peptide bond, so it is not correct. Now, so question number 6 will go, which mass photospectrometry, sorry, mass spectrophotometry, you will find or you will have to use a matrix. So their answer is MALDI. This is again directly from the lectures. Then in question number 7, the correct answer here is option number B. Okay, so if you look at my slide, you will see there is a governing equation that I have found from the internet. And in this equation, you will see that the mass of the ion is directly proportional to D. If you see this particular equation, means more heavier the ion, more time it will take to reach the detector. So it is directly proportional, not inversely proportional. Okay, so this question will also be rectified. Then go to question number 8. This is how the diagram representation is. The heavier, the larger ions, they are at a farther distance from the detector, whereas the smaller ions, they are moving ahead. Okay, and this is the governing equation which will help you calculate the time of flight in this kind of an assay. Now, question number 8, go to question number 8. So there you have to answer which of these assay can help you identify apoptosis. Okay, so there is MTT, LDH assay, scratch test, and biocompatibility. So there is no test that is called biocompatibility. I think you guys have understood so far. So option number D is obviously not going to be correct. Then scratch test is helps you to analyze whether cell is able to migrate or not. Whereas MTT, it helps you to analyze the cell proliferation rate, cell metabolism, cell viability as well. So MTT is also not correct. Whereas if the apoptosis has caused uh, alteration in cell membrane permeability, then the LDH assay can help you detect that. And which is why here the most appropriate answer is option number B. Now question number 9, if you see, complement activation does not lead to. So complement activation, if you again go back to the lectures, you will know what is complement activation, and directly you can understand it is option number C. Blood clotting, it does not causes blood clotting. For that, there is coagulation pathway. And then option number and last question, the scratch test is performed to study. Very simple, it is used to study cell migration. So I've already discussed this in today's class as well. So these are the answers for week 4 assignment. Okay. And I hope you guys did well. So if you guys have any additional queries after today's session, you guys can post it in the course forum. I'll be happy to take a look at it and answer it. If not, we will discuss it in the next week, Saturday, and we'll meet at 4 p.m. All right. Next week, we'll discuss the metallic biomaterials, polymers, basically the content of week 5. Okay. So if anyone has any query or additional thing to say, you guys can say it now. Otherwise, I'm done from my side. Anyone has any query? No queries? Everyone was able to understand whatever was discussed today? Did you find anything very difficult to understand? Okay, one person gave a thumbs up. What about others? Okay, another thumbs up I got. All right. Okay, so then I'll assume that you guys have understood the content that I discussed today. But if you have any query, feel free to come prepared and you can ask me directly in next week's session. Okay, go through week 5 content again. Okay, and we will definitely have a revision class towards the end. But before that also, if you have any query related to any of the week's content, you guys can definitely ask me. All right then,
Ma'am,
I'll
the
yes.
attendance will be considered for your grading. The attendance.
Um,
Hello.
so you guys have the grading policy, right, of the course. So it is clearly written what was so for the record, I have to like keep track of the attendance because the NPTEL wants to know how many of the students are attending these sessions. So it's also a reflection on my performance, how much I'm able to clarify your doubts. If more students are joining, it means like more people are interested in these problem solving sessions. If not, then maybe they will stop doing these sessions. Like it's not a remark on you or how you're performing. It's more of an evaluation from us who are attending or who are conducting these sessions. Okay, who are like we are PhD scholars after all, and um, we take out time from our schedule to prepare these sessions for you guys. So is it helpful for you? If yes, then you guys should attend. If not, then you guys shouldn't attend. So it's a more of a evaluation for us and the NPTEL team. It is not an evaluation of your performance. But anyways, you guys know, right? So how much weightage the assignments have, how much weightage your exam will have. So you guys already know this. So it is already written in the course about page. You can go and read. So I don't think anything is written on about attendance. So feel, but if you have any query, which is why you are asked to join this session. If you have no queries, then it's totally fine. Don't attend. All right.
Ma'am, I think most of the students are not joining is because of maybe they, I mean, like as of me, I have some work. I actually wanted to join most of the sections, so that's the reason. It's not because we're not interested or anything.
So you anyway will get the recorded lectures, so you guys can definitely view it afterwards. So so students join, no, as a person who is delivering this content, it helps me to understand how well I'm delivering, how well I'm able to explain these concepts. So I'm also a PhD scholar, and my prospective job will be to become a teacher. So I'm also trying to hone this skill of teaching. So it makes me feel good that people are joining and they're asking questions and I'm able to answer them. So yeah, it's good if you guys attend. Okay. And definitely, if you have no queries, don't attend. It's like it is up to you how important you think it is. Okay. Anything else?
Yes, ma'am.
Yeah. Okay. Anyone else have any remark or any query to ask? All right then. Let me then end today's meeting. I hope you guys have understood whatever was discussed today, and I'll see you guys next week on Saturday from 4 p.m. Okay, bye, guys.
Thank you, ma'am.
Yeah.